Today we're going to be discussing the association of the infant gut microbiome with early childhood neurodevelopmental outcomes. The purpose of this presentation is to discuss whether the gut microbiome in early childhood has the ability to predict neurocognitive skills later in life. The gut microbiome and the gut-brain axis are fascinating areas of research which demonstrate how bacteria, viruses, and other microbes living in our bodies have the ability to affect our behavior and our brain development. The authors of this study were interested in how this microbiome has the potential to affect a child's neurodevelopmental skills. They collected stool samples from over 300 infants aging from 3 to 6 months old and observed whether there was a specific gut pattern that seemed to influence ASQ3 or cognitive skill scores at the age of 3 years old. The study determined that there were higher levels of Clostridiales and Bacteroides in certain infant stool that seemed to have the greatest effect on development later in life. The results likewise demonstrated that specific microbe concentration may matter more than microbial diversity in itself in terms of childhood development. This study was an ancillary study, meaning that it was done in addition to data that had been collected at a previous time. The authors pulled from the VDAART study, or the Vitamin D Antenatal Asthma Reduction Trial, which aimed to measure how the administration of vitamin D can impact asthma in infants. So let's put all of this information into context. Animal research has shown us for a long time that the gut-brain axis may be incredibly important in altering behavior, anxiety, neurodevelopmental needs, and cognition. However, not much of this research has been conducted in humans. It's incredibly important to begin looking at this because before this paper, many studies were small and they were focused on animals, or they were looking at older children and adults rather than infants themselves. This study approaches this subject from a research point of view with young kids and follows children from the beginning of development to see how their gut bacteria might relate to early cognitive development. So you may be wondering why this question is so important. Well, if we can identify early microbial markers in infants linked to their neurodevelopmental outcome, we might one day be able to screen children who might be at risk for a developmental delay or for any sort of neurocognitive delay. The rates of neurodivergence are currently increasing, and we don't know why, but we do know that an advancement in this field may allow us to design simple and low-cost treatment plans and interventions such as probiotics, maternal dietary changes, or even develop microbe therapies. The broader implication of this is societal. Such a development could open a whole new world of early childhood treatment and could potentially reduce the lifelong impacts of neurocognitive delays. So simply put, the purpose of this article was to ask the question, does the gut microbiome at 3 to 6 months of age predict neurocognitive ability at 3 years? The authors simply hypothesized there would be a correlation to some extent, and that the specific gut bacteria may be linked to developmental skills like motor coordination, social behaviors, and communication. Now let's discuss how the authors went about answering this question. First, they used participants from the VDAART trial and pulled data from 309 previously tested infants. When the infants were between the age of 3 and 6 months, parents at home collected stool samples and had them sent to the lab for analysis. 16S rRNA sequencing was used to identify which bacteria were present in the stool samples and to analyze the concentrations of these specific bacteria for each infant. The authors then used a factor analysis method to group the bacteria into coabundance factors, which allowed them to look at patterns of bacteria within the stool samples instead of just individual species. Then, when the children turned 3 years old, their parents completed an Ages and Stages questionnaire or an ASQ3 test, which was used to measure different childhood development categories across 5 domains, including gross motor skills, fine motor skills, problem-solving, communication, and personal social skills. Once this was completed, the scientists analyzed the results from the stool samples in comparison to the ASQ3 results to note any trends. When the infants chosen were between 3 and 6 months old, the parents collected stool samples at home and had them sent to the lab for analysis. These samples were then analyzed using 16S rRNA sequencing, and bacterial concentrations within the stool were observed. The authors used a factor analysis method to group bacteria into co-abundance factors. What this term means is that they weren't just observing a specific bacterium, they were looking at patterns of bacteria and grouping them together based on what tended to rise and fall. Notably, they chose to focus on Bacteroides dominant and Clostridiales dominant bacterium, as those were the two factors that were deemed to be statistically significant based on a 0.05 significance level. When the children turned 3 years old, their parents answered the ASQ3 questionnaire, which asked several questions to assess their skills in numerous categories, including gross motor, fine motor, problem-solving ability, communication, and personal and social skills. Each item in the ASQ3 test was scored on a scale that was ranked on a 0 to 10 point basis. Yes was 10 points, sometimes was 5 points, and not yet was 0 points. This allowed for a total of 60 points to be collected throughout the test. Children were then assessed on a point basis based on where their parents believed they were at developmentally. Now let's take a look at the massive table on the right and talk about the two main patterns that stood out to the scientists following this experiment. They found that higher levels of Clostridiales bacteria in the infant gut at 3 to 6 months was associated with lower communication and social scores when they were 3. This implies that there is a correlation between Clostridiales and social interaction challenges and problems with speech development. They also found a high level of Bacteroides bacteria associated with lower fine motor skills, which implies that there is an association between Bacteroides bacteria and the ability to grasp objects, use fingers precisely, draw, or do anything that requires fine motor ability. First, let's discuss the Clostridiales dominant factor results. The authors found that a higher relative abundance of this factor, factor 1, at the age of 3 to 6 months was negatively correlated with communication and social skills. This implies an association between high levels of this bacteria and lower scores on these areas when they turned 3 years old. We can see in the data that as there was an increase of factor 1, there was a statistically significant decrease in communication scores and in personal and social skills. It's worth looking at the p-value here because the p-value is what determines statistical significance, and for the purpose of this study, a p-value of 0.05 was used. Because the results are 0.05 or under, this deems them statistically significant. Also worth noting, the beta coefficient for this communication was negative, which means that the communication and personal and social skills and factor 1 bacterial presence will have an inverse relationship, or they will move in opposite directions to each other. As one increases, the other decreases. The personal and social skills p-value is surprisingly small and indicates that it is highly unlikely that there is not a correlation between these two variables. Additionally, the Bacteroides bacterial presence, or factor 3, was assessed, and it was determined that a high abundance of Bacteroides at 3 to 6 months old was statistically associated with lower fine motor skills at the age of 3 years. We can see in the highlighted section the p-value was 0.01, suggesting that there was a high correlation between these. We can also see that, like with the last slide, the beta coefficient is negative. Unlike the Clostridiales bacteria, Bacteroides only seemed to correlate significantly to one factor. It's worth mentioning that the association remains significant regardless of potential confounders like maternal education, the mode of delivery, and breastfeeding. There was an association present between fine motor skills and Bacteroides. To support the results determined by the co-abundance factors, the authors ran a separate type of analysis called the multivariate association with linear models analysis, or the MASLIN. This allowed for them to check whether individual bacteria were associated with ASQ3 scores in the same pattern, not just a bacterial genus. Before I continue, it is worth noting that the image on the slide was not pulled from the study, as one limitation to the study was that the researchers did not provide their MASLIN analysis. I included this image to demonstrate what a MASLIN can look like and to show you how they are frequently formatted. For example, they test for abundance and prevalence. They show several different bacterial types and species. They also demonstrate the covariates and the beta coefficients. In the case of this study, it was reported in the results section that the MASLIN findings were consistent with the factor findings, mostly. In terms of communication skills, the MASLIN demonstrated that children with better communication scores had lower abundance of Clostridiales, just like in the co-abundance factor analysis, but the MASLIN was able to provide more specific information. For example, the genus Ruminococcus from the Lachnospiraceae family was deemed to be in lower abundance with a coefficient of negative 0.065 and a p-value of 0.04, indicating statistical significance. In the MASLIN scan, there were 6 top hits, and for personal and social skills, 3 of those showed the taxa with Clostridiales were less abundant in children with stronger social development. This also supports the findings of the co-abundance factor analysis. And in terms of fine motor skills, the MASLIN analysis did not actually show the same Bacteroides genera from the co-abundance factor. Where the results differed was that in the case of fine motor skills, the MASLIN didn't actually demonstrate the Bacteroides results that were found from the co-abundance factor analysis. The test reported a different relation between lower levels of Enterobacteriaceae and higher levels of fine motor skills. Because the Bacteroides is a genus and the MASLIN is designed to pull out individual bacteria, it wasn't reported. But what was reported was that Klebsiella had a coefficient of negative 0.033 with a p-value at an astonishingly low 0.001. This indicates extreme statistical significance that a simple co-abundance factor test would not be able to pick up. For this reason, it was very beneficial that the scientists ran a MASLIN test because it reinforced the broader factor results, but also demonstrated further detail about which specific families might be driving these individual patterns. It's also worth mentioning which variables and results were not significant. So the MASLIN didn't actually separate Bacteroides at the genus level to deem statistical significance for fine motor skills. So what this means is that the MASLIN model was more focused on specific bacterial taxa. So it instead found that Enterobacteriaceae was significant, but did not determine a link between Bacteroides and fine motor skills. This means that according to the MASLIN test, that link between factor 3 and fine motor skills was not significant. The results also suggest that the overall microbiome is not statistically significant, and that instead it is the individual bacteria that mean more than the microbial diversity. Likewise, other ASQ3 factors like factor 2 and factor 4 were never deemed to be statistically significant or have any sort of significant difference. Overall, these non-significant results also highlight that not all aspects of the gut microbiome do predict neurodevelopment in the same way. So it indicates that while some may be indicative of neurocognitive ability, not all will be. It's specific bacterial groups, not the diversity in itself, that shows the significant link. And those effects appear to be domain specific. So they appear to depend on the bacterial domain they are a part of. Notably, the authors also observed whether maternal vitamin D supplementation during pregnancy may have impacted the infant's gut microbiome at birth. They discovered that moms who took vitamin D when they were pregnant as a result of the VDAART trial had babies who had lower levels of potentially harmful bacteria. This implies that there is a correlation between maternal vitamin D supplementation and decreased infant gut bacteria. One of the most interesting results of this study was that overall microbiome diversity didn't seem to cause any sort of difference in neurodevelopmental outcomes throughout the results. However, there were differences noted between factors, which implies that microbial diversity in itself is not what influences neurodevelopment, but specific groups of bacteria being in higher concentrations may cause a difference. It is worth noting that looking at the table, the results don't seem to vary that significantly, which is to be expected because brain development is likely caused by a variety of different variables, not just the gut microbiome. However, these results were statistically significant. It's important to consider the implications of these results not only in the context of this study, but from a larger societal standpoint as well. The key takeaway is that the infant gut microbiome may be an early indicator or even potentially a factor in how a child develops socially and practically. The results of this study open the doors for ideas like early microbiome screening for infants, probiotic treatment for mothers to be passed to the child through the breast milk and the umbilical cord, and therapies that target prevention. It's worth noting that the study also implies that maternal nutrition is very important and that maternal vitamin D supplements can actually be protective for infants against harmful bacteria. While this doesn't directly answer the question of this study, it does show how crucial the microbiome is for childhood development. There are various strengths as well as limitations to this study, including a large sample size for this kind of research. The study polled from 309 individuals, which is a massive research population and allows for a lot of samples to be collected. Likewise, the study used verified methods such as 16S rRNA sequencing and ASQ3 testing and made note of multiple factors that may have impacted bacterial concentrations, for example, vitamin D intake. However, it is worth noting that only one stool sample was collected from each infant, so we don't know if the microbiome could have changed over time or if it could have been a situational result as opposed to a consistent one. Likewise, 16S rRNA sequencing was only completed one time for each sample, and it is always the case that trials lead to more accurate results and more reliable results. It's also worth noting that the home environment was not discussed, so we don't know fully if there were any outside factors that may have been contributing to the child's development. And the ASQ3 testing was an established method, but was reported by the parents and therefore could have some sort of implicit bias. Regardless of these limitations, however, this study is an incredibly important step towards understanding the gut-brain connection in infants. However, we do need more tests to figure out causality as opposed to simply correlation. So, as a reminder, this study assessed correlations between 16S rRNA sequencing results for 3 to 6 month old infants and their corresponding ASQ3 scores at 3 years old. This study provides evidence that specific patterns in the infant gut microbiome may influence development by the age of 3 years old. It also suggests There is great potential for future screening and early intervention for infants in the future. This is so incredibly important because it could help us identify and support children who are at risk for neurodevelopmental challenges later in life when they're young, using safe, inexpensive, and non-invasive approaches to make their lives happier and healthier. However, more research is absolutely needed before we can make this kind of clinical recommendation. So before we finish, I wanted to leave you all with a few questions to consider that I believe are incredibly applicable to this research study and to potential future studies. Firstly, should microbiome screening be mandatory at birth? And what are the ethical considerations of this, as well as of microbiome modifications in infants? Could environmental factors, for example, a happy home, influence the infant microbiome? To what extent does the infant microbiome impact mental health in addition to the skills monitored by the ASQ3 test? And finally, what possible research would be informative in response to this study? How can we build on this? What more can we do? Thank you so much for your time and your attention. I would absolutely love to hear your thoughts about this study in the comments, and I can't wait to read over them. Thank you, guys.